You may have heard of the Human Genome Project as a major scientific milestone. The Human Genome Project was run from 1990 to 2003 and set out to identify, map, and sequence all of the genes in the human genome. The project was considered complete in 2003, but it only contained roughly 92% of the actual genetic information it sought to obtain. This is because it relied on a DNA sequencing method that involved short-read sequencing. In short-read sequencing, DNA is broken up into smaller fragments for processing, but at the expense of some genetic information. Long-read sequencing, on the other hand, does not require DNA to be broken up. Nanopores fall into this category. Development of nanopores began in the late 1980s and continued on through the mid-2000s when the technology was licensed by a company called Oxford Nanopore Technologies. Just last year, in 2022, the gapless sequencing of the human genome was officially complete thanks to the nanopore sequencing mechanisms produced by Oxford Nanopore and run by the Telomere-to-Telomere Consortium. But how does this technology really work? On an individual scale, nanopores for DNA sequencing require some form of electrically resistant membrane. This could be a lipid bilayer, or, in the case of Oxford Nanopore, a polymer membrane. A pore-forming agent could be introduced to produce a protein pore through the membrane. Some commonly chosen proteins include alpha-hemolysin and Mycobacterium smegmatis porin A, or MSPA. This pore usually has an internal diameter between 1 and 2.5 nanometers. The pore should be the only pathway between the two sides of the membrane, which is situated in an ionic solution. When a voltage is applied across the membrane, an ionic current should be produced through the nanopore. This current can be measured by an ammeter. But where does the DNA sequencing come in? Well, DNA is negatively charged. If the applied voltage runs from negative potential on the cis side of the nanopore to positive on the trans side, DNA will be driven through the nanopore. To sequence DNA, a motor protein is bound to the cis side of the nanopore. Polymerase is often used for this. This motor protein binds to double-stranded DNA inserted into the cis side of the membrane and aids in unzipping it to form single-stranded DNA. As the voltage is applied and an ionic current is produced, the single-stranded DNA is ratcheted through the nanopore by the motor protein. This causes the current measured by the ammeter to change as the different nitrogenous bases cause different disruptions to the current based on their size and structure as they pass through the pore. Several nucleotides are usually within the current reading range at any given time, but machine learning algorithms can then be used to decode the current fluctuations recorded by the ammeter to reveal the order of the bases in the DNA. In 2014, Oxford Nanopore Technologies began selling their first device, the MinION, that consists of 2,048 nanopores. As of 2023, its base read accuracy is up to close to 99%. The device is small enough to be held in your hand and is powered through a USB-C port on a computer. It is also sold for just $1,000. This greatly lowered the financial barrier to entry for genetic research, as old DNA sequencing systems cost on the order of $100,000. Oxford Nanopore has since released the GridION with the power of 5 MinIONs, as well as the PromethION with the power of 40 MinIONs. In 2022, the PromethION set the record for fastest sequencing of a human genome with a time of just 5 hours. Because the MinION can be powered by a laptop, it has been taken to a wide variety of applications around the world. To date, the MinION has been used for fieldwork and genetic testing in developing countries, as well as to study life's origins in remote volcanoes. Nanopore sequencing also played a major role in our understanding of COVID-19, which aided in vaccine development. The MinION has even been brought to the International Space Station to study the influence of space on both the properties of microbes as well as the human immune system in preparation for a future flight to Mars. Nanopore sequencing has opened up the study of DNA and RNA to many more researchers around the world who would not have previously been able to participate in genetic research. Thanks to its speed, portability, and accessibility, nanopore sequencing should bring even more groundbreaking research in the future.